First off, what is mitosis? Mitosis happens in somatic cells, which are any cell of a living organism other than the reproductive cells. There are 4 stages to mitosis, not including interphase, and the purpose of mitosis is to make more cells or cellular proliferation. The chromosome number stays the same and there is no genetic variation in mitosis. Mitosis produces 2 daughter cells. So here are the different phases of mitosis, which are prophase, metaphase, anaphase, telophase, and cytokinesis, and a quick little diagram of my models and the different parts that are in the cell, which are the cell wall, chromosomes, spindle fibers, and the centrosome. Interphase is the growing and developing phase of cells, and it is very important. And then we move on to prophase, which is where the centrosome splits in 2 and the nucleus starts to dissolve. And in metaphase, the nucleus is completely gone and the spindle fibers form and hold the chromosomes in the middle of the cell. In anaphase, the spindle fibers pull the chromosomes to their respective sides, which is the 2 opposite sides of the cell. In telophase, 2 nuclei form around the separated chromosomes, and you start to see that they're almost their own cells. And then in cytokinesis, the cell splits into 2, and you are left with the daughter cells. What is meiosis? The purpose of meiosis is sexual reproduction. The chromosome number is halved in each cell, and there is increased genetic variation. Meiosis produces 4 haploid daughter cells. So, the different stages of meiosis are as follows: prophase 1, metaphase 1, anaphase 1, telophase 1, and then cytokinesis, which is where the cell splits, and then the same cycle again for a second time, which will happen with half the amount of chromosomes. Because since meiosis produces 4 cells, this happens to multiple cells at the same time to create the 4 daughter cells. The interphase phase is the same, where it is just growing and developing. In prophase, it's also again pretty the same, but in metaphase, the chromosomes cross over bits of themselves to create more genetically diverse specimens. And then in anaphase, yet again, they are pulled apart to the separate sides, and then in telophase, they are again on their respective sides. So cytokinesis splits the cell, and then it happens again with prophase 2, and you'll notice that this time there are only 2 chromosomes left in the cell. So when those get split, there is half the amount to create gametes, which are the sex-reproducing haploid cells. So what is the same? Both processes aim to produce new cells. The different stages are relatively similar, and they both start with a single parent cell.